Hello, everyone. Thank you for joining us. Welcome to the Avexia webinar series. Our topic for today is multi-cancer early detection, screening with liquid biopsy. My name is Christopher Chu. I am the Director of Marketing for Avexia Diagnostics and will be your host for this webinar. Today, I have the distinct pleasure of being joined by an exciting guest, Dr. Richard Burnett, who will be our presenter for this webinar. After completing his undergraduate studies in microbiology and immunology, Dr. Burnett earned his medical degree from the University of Arizona and completed his anatomic and clinical pathology residency at the Arizona College of Medicine. Following his fellowship training in cutaneous pathology at Jefferson in Philadelphia and active duty in the U.S. Air Force in Texas, Dr. Burnett served as a medical director for a large regional laboratory, a partner in large private practice, bootstrapped his successful laboratory, and serves as a leader in sonic healthcare. Over his career with academic appointments at the Mayo Clinic, the University of Arizona, and now Midwestern University, he has trained and educated scores of medical students, residents, and colleagues. Dr. Burnett now serves as COO of Precision Epigenomics. He hopes to make his greatest professional impact working with their extraordinary team to improve health through earlier identification of cancer to support clinicians in better care for their patients. Without further delay, I will now turn the webinar over to Dr. Burnett.
Thanks, Chris, and thanks to everyone for joining me today. I just wanted to tell you a little bit about multi-cancer detection and liquid biopsy solutions. Again, I'm Rich Burnett. I'm a pathologist with Precision Epigenomics. So today we're going to talk not only about cancer. It's an enormous problem, more than 2 million new diagnoses in the USA alone, more than 600,000 deaths per year. Globally, it's obviously a much bigger problem, nearly 10 million deaths per year. It's extraordinarily expensive. There's a lot of suffering associated with cancer. The folks that are dying from cancer are the folks that tend to be diagnosed late or late detection. If you look at the table on the left, you can see that while cancer is obviously a huge problem, the real calamity, the catastrophe is delayed detection. So delayed detection is the real problem that we're facing. When cancer is diagnosed relatively early in stage 1 or stage 2, there can actually be pretty favorable outcomes. With late detection, stages 3 and 4, the survival drops dramatically, the suffering goes up, the cost associated with management goes up. So really a big problem. So we want to get these cancers detected early when treatment is more likely to be successful for a complete cure. The problem is that only 4 cancers today, each with its own detection or screening test, are available widely. And that's really a problem because they tend to underperform. So cancer screening in the US, what is it we're trying to accomplish? So we want to be able to detect cancer early. And the idea of detecting it early in stages 1 and 2 is that the chances that somebody who's actually getting screened will have better survival than somebody who's not getting screened. So that's obviously a goal. But the flip side of that is that the screening test itself has to be effective, and it can't induce more harm than good. In other words, we can't have people stop getting their routine standard of care screening. We can't have so many false positives that people are getting very expensive workups to look for a cancer that's not even there. We can't have bad sensitivity or too many false negatives, missing cancers that are there. So those are the goals of screening in the US. You can see here, these are the cancers that we're currently screening for. So 4 cancers, 3 of them are in the top 10. Cervical cancer, the Pap smear, Pap testing has been extraordinarily effective. It used to be a horrible cancer with lots and lots of incidents, new cases being diagnosed. With Pap testing, we've actually done a great job of making it much less common. But of the big cancers that remain, we're really looking at breast, colorectal, lung. But these, again, they tend to underperform as a rule. They're not bending the curve. And too many cancers are being diagnosed late. So again, if you look at this is from the American Cancer Society. The cancers on the top, again, feel free to pause at any point if you want to look at this a little closer. But the cancers on the top are the new diagnoses that we're seeing. The cancers on the bottom are the fatalities each year, cancers that are actually killing people every year with men and women. So lung, breast, colon, You see PAP didn't make it here. Uterine cervix is not one of the top 10, but that would be in here as well. Prostate's a special consideration. We were checking PSAs on lots and lots of men. We got into a situation where we were probably overdiagnosing prostate cancer, detecting very small cancers that the patient probably would have been fine living with for many, many years. So prostate is a little bit more of a complicated question, and it's not one of the four. It's routinely recommended screening broadly. It needs to be a special conversation between the healthcare provider and the patient. So what are multi-cancer detection tests? Well, it's pretty straightforward, but the idea of this, currently we have one screening test for one particular cancer type. But what if we had one test that could detect multiple cancer types? And that's what a multi-cancer detection test is. The idea is that we're going to take a peripheral blood specimen, or it's called a liquid biopsy by some people, and look at that specimen and see if we're able to detect different cancer types aside from the ones that we're already doing routine standard of care screening for. So these are designed to be complementary to the existing ones, even though many of these multi-cancer detection tests will detect colon or breast. Nobody is suggesting that they get rid of routine colonoscopy or fecal test or mammography looking for breast cancer. Nobody's suggesting that. But these are being pursued by big academic institutions, big companies, small companies all across the world. And it's an extraordinarily exciting time to be in this space. And we're grateful to be part of it. These tests are still early, though. So these are not FDA approved. None of the multi-cancer detection tests available on the market are FDA approved. There are some single cancer tests that were recently approved that are available on the market, but again, not multi-cancer detection. Because of that, most of these are not going to be paid for by third-party payers. So Medicare is probably not going to pay. Blue Cross Blue Shield is not going to pay. These are cash pay for these multi-cancer detection tests. So let's talk about the epigenome. We need to have some context so that you understand a little bit how this test works. So I think we all remember that DNA goes to RNA, goes to proteins as a rule. That's kind of the central dogma of biology. But there's more to it than that. All of the cells in your body have essentially the same DNA. And we know intuitively that a skin cell is different than a gut cell, is different than a brain cell, is different than a heart cell. And that has to do with the epigenome. So the epigenome can be modified to turn different parts of all that DNA on or off. So for example, in some cells, we want this piece of DNA to be expressed into RNA and be turned into proteins. But in a different kind of cell, that's not helpful at all. We don't want that at all. So the epigenome is one way that regulates the cellular differentiation. If we look at a stem cell, again, all of that DNA is in there. But what happens is the epigenome, by methylating different parts of it, will close down the DNA, and that part of the DNA doesn't get expressed. For example, if you're going to be a chondrocyte or an osteoblast or a keratinocyte, you're going to want to have a certain set of genes open. But if you're going to be a white blood cell, you'll have other ones closed off. And the methylation of the DNA is one of the ways that the DNA is controlled to turn off and turn on different parts of that DNA that gives us different differentiation or different cell types. Now, in normal development, things work wonderfully. But in cancer, sometimes the epigenome gets a little bit out of sorts, and things that are supposed to be on get turned off, and things that are off get turned on. So we looked at this methylation signal as a real way to determine what's going on with these cancers. So we looked at an enormous number of cancer types. We looked at differentially methylated regions, areas that are abnormally methylated in cancers, but aren't abnormally methylated in normal tissues, whether it's heart or lung or brain or colon. I mean, we wanted to find these areas and create probes that would detect those abnormal areas if they're present, but then not bind and not amplify if they weren't present. And then finally, we prepared a software classifier to look at all those outputs and stratify the results so that we'll know if abnormal methylation signals that are typical of cancer are present, or if those abnormal methylation signals are not present, and it's very, very unlikely the patient has malignancy. So we came up with the EPISeek. It's our multi-cancer detection assay. And just to explain how it works, if you look at the image on the left, you can see a very small lung cancer, for example. It could be anywhere in the body. But as the cancer grows, there are multiplication of cells. They're growing, but it's not linear. Some of the cells are dying. And when the abnormally methylated cancer cells die, they release that abnormally methylated DNA, tiny little fragments that are floating around in the peripheral blood. And that's called circulating tumor DNA, which is one kind of cell-free DNA, or DNA that's not in the cell where it's supposed to be, it's outside floating around. Well, when it's outside floating around, it's going through the blood system, waiting to be taken up and destroyed. But while it's floating around, we're able to do a peripheral blood draw and take a simple specimen of blood and determine if there are fragments of abnormally methylated DNA in there. We've developed these patented probes, and by using these probes that detect cancer signals, we're able to detect if cancers are there. Initially, the image on the right shows the cancer types that we were targeting. We said, let's just go after some of these big top 10 type cancers. Let's go after these that are killing a lot of folks. Lo and behold, when we did additional studies, we found out that these abnormally methylated regions not only picked up these cancers, but a total of 24 roughly currently, but it could be more, but certainly more than 20 cancers that are, causing enormous suffering and loss of life. So if we look at it now, if we look at what EPISeek can detect, these are all the different cancer types that EPISeek has detected using plasma as a specimen type. So you can see all the big ones. Leukemia, to be frank, can be picked up on peripheral blood semen pretty easily. We don't really need a molecular test for it. But the other cancer types you can see, all those were the only multi-cancer detection test that actually detects brain cancer, what's called glioma or glioblastoma. So that's unique to us. And you can see some of the other cancer types in the right margin there that shows some of the other cancer types we detected. So many, many cancer types. So what I want to make clear, though, is that our test performs better with some cancer types versus other cancer types. So if we look at the cancers on the far right, the stomach, the cervical, and again, you might want to pause the image or the video just to take a look at it. You can see that the sensitivity is really good for those. And let's just pick bladder cancer, for example. So 75%. What does that mean? That means if we tested 100 patients with bladder cancer, our test would correctly identify 75 out of those 100. We would miss 25 of them. But again, I think what's important to understand is currently the screening that we have in place now is 0%. So 0% for some of these aggressive cancer types, and we're getting 75% sensitivity. So while you might want it to be 100%, and I do too, the reality is that it's not perfect. There are some early cancers that it won't detect. There are certain cancer types that it's less likely to detect. If we look at prostate, for example, prostate cancer for us and for other people that are doing multi-cancer detection tests with liquid biopsy, it's very difficult for some reasons that aren't clear to us. When prostate cancer is growing, it doesn't tend to release a lot of circulating tumor DNA out into the blood for our test to detect. Or if it does release it, it is somehow cleared very quickly or modified somehow so that it's very difficult to detect. But again, I just wanted to give you an overview and make sure you understand that it performs better for some cancer types than other cancer types. This is an overview of the different stages. And you recall that stages has to do, is it an early cancer or a late cancer? For our purposes, we put stages 1 and 2 together. So that second paired column that you see there, that's stages 1 and 2. And again, you might pause if you want to look closer. Stages 3 and 4 are to the right of that. I don't think anybody would consider stage 4 early cancer detection. That means it's widely metastasized throughout the body. Stage 3 means that it's gone to the nearby lymph nodes, not necessarily a sentinel lymph node, but it's spread out of the organ at least to 1 or 2 lymph nodes nearby. Still operable, still can change the outcomes, but stage 1 and 2 are probably the most important. What we've illustrated here are 2 different color bars, and this is to show our full disclosure, so you see everything. The blue is all the cancers, and these are some of the cancers that include thyroid cancer, myeloma cancer, or plasma cell cancer. Some of these are more indolent, they're slow growing. Early detection probably isn't as big a deal for these cancer types as it is for some of the more aggressive cancer types. And by aggressive cancer types, I mean the ones that have the worst 5-year survivals. So the cancers that have the worst 5-year survivals are ones that are growing quickly, they move quickly, and if they're not diagnosed early and treated effectively initially, they tend to have pretty bad outcomes. And we further refined it by saying, which are the ones that don't have current commercially available screening tests? For example, we did not include breast cancer because breast cancer has a mammography that's pretty effective. We did not include colon cancer, same thing. There's endoscopy to look directly. There are fecal tests to look for colon cancer. But these are cancer types listed there that you can see that we don't have widely accepted, widely adopted screening tests. And you can see that for those kinds of aggressive cancers, for stage 1 and 2, we're about 57%. And again, keep in mind, currently it's 0%. So 57% is actually quite good. For stage 3, it goes up to 73%. And again, stage 4, less important. The disease is already spread, and diagnosing it, a month or a week earlier really is probably not as important as diagnosing it in stage 1 or stage 2. So I want to, again, emphasize that there are many unknowns about multi-cancer screening. The NIH and the NCI in particular have started a long study. It should take 6 or 8 years to try to suss this out, to figure out, what are some of the things that we don't know about multi-cancer detection testing. What happens if somebody has an abnormal cancer signal detected type report, but the cancer is not found? Do we keep following them more closely, or do we not follow them as closely? Is this going to make people get relaxed about doing their standard of care screening? Are they going to stop getting mammography? Are they going to stop getting colonoscopy? These are real questions that I want you to at least consider as you're about to start using multi-cancer detection in your practice. So if you choose to do it, and I personally think it's a good idea, if you choose to do it, here is the EPISeek specimen collection kit. The specimen collection kit is a cardboard box, and within it is a polystyrene or a styrofoam type box that's there for protecting the specimen while it's in transit. Every specimen doesn't always get handled by, with kid gloves. Sometimes they get thrown. So we have that foam box in there to protect those test tubes that are holding the blood specimen, but also for thermal control. So the Precision Epigenomics Laboratory is in Tucson, Arizona. And if a specimen is shipping anytime between May and October, it's hot in Tucson, Arizona. So we want to be able to protect the control the thermal temperature inside of that container while it's in transit. By the same token, in the wintertime, the northern neighbors get pretty cold, and we certainly don't want that blood specimen to freeze en route to our laboratory. So in that case, the styrofoam box will help keep the temperature above freezing in case it sits out on a tarmac somewhere in the north. So inside the kit, inside the polystyrene, you'll see there's collection instructions, there are shipping instructions, there's a saddle gel pack next to the instructions. That gel pack can be kept at room temperature. Again, we're just trying to provide some thermal mass for the blood tubes. You can refrigerate it if it happens to be summertime, but again, not necessary. Just please don't freeze it. We don't want the collection tubes to freeze. Next to those little gel packs, you can see 2 Streck tubes. Streck tubes are not routinely stocked by most laboratories or hospitals. That's why we provide these particular specimen collection tubes. You can see there's a human biologic specimen bag, a little bit of absorbent material, and finally there's a FedEx return service. For our domestic specimens, the entire kit will be put back into that cardboard box. The cardboard box will go into that FedEx mailer for domestic, and it'll make it to our laboratory the next morning for processing. I think it's also important to understand that we want to get these things processed pretty quickly within 48 hours after the specimen is collected. So no late afternoon collections and the FedEx guy has already come. Try to do these earlier in the day before the FedEx is picked up. Don't do these on Thursdays and Fridays, because invariably there'll be a delay, and then it won't come Friday. We won't see it till Monday, and it'll be outside the 48-hour window. So please try to collect these Monday through Wednesdays in the mornings. Get them to our laboratory, ideally the next day. We really want to turn these around quickly for you, because that's one of the advantages of our platform. It's much faster than next-gen sequencing, which is a different platform. So, I think that's really probably the most important stuff about the specimen handling is that collection time, getting it to us, draws on Monday through Wednesday. Again, get it in FedEx the same day. Try not to let it sit overnight and lose a day for no good reason. Let's talk about the reports that you're going to get. So the cancer signal not detected, this is by far the most common report that we issue and that you're most likely to get, especially if you're truly dealing with screening patients. In other words, no signs and no symptoms of cancer. We say cancer signal not detected. Now, again, it's important to recognize that this test is not 100% sensitive for particular cancer types and for different stages. So because a patient gets this result, we can't say with complete certainty that they don't have cancer, but the negative predictive value is 99%. So they most likely do not have cancer when they get this result. So again, I think it's probably the most important thing I want to add on this as well is that the patient still may develop cancer. They should continue with their routine recommended screening. And that's something that's important that you emphasize to your patients if they're considering this particular assay. Now, this is much less common, but this is the abnormal methylation signal detected report. We don't say cancer signal detected because the positive predictive value is less than 50%. It's 40%. So more often than not, if somebody has a report that says abnormal methylation signal detected, while they're certainly at risk, they're much more likely to have cancer than somebody who has a negative. This is not a diagnostic test. This does not mean the patient's been diagnosed with cancer. All this means is that this patient is at higher risk. There's some abnormal signals present, and they need to get in and be followed up with an additional study. Currently, additional studies might be PET-CT scan, other kinds of imaging, laboratory tests, things along those lines. But again, the most important thing I want to emphasize is we don't put in the report cancer signal detected because it may not be cancer, and we don't want people to think they've been diagnosed with cancer. We say abnormal methylation signal detected. When this happens, you will be getting a call from one of us, one of the pathologists in our laboratory. We call for all of our positives and just discuss it with the healthcare provider who ordered the test. Find out if there's any additional history, help the healthcare provider make some next steps, understand how to manage this particular result, what the next steps would be for the patient so that nobody's out in the cold wondering, what do I do with this result? How do I handle this? We're here for you. And even for negative results, if you have questions, feel free to give us a call. So that's it. I want to thank Avexia. I want to thank you all for listening to the presentation. I put in quite a bit of information. Again, if you want to listen to it again and hit pause from time to time, feel free. But at this point, I'll turn it back over to Chris. And just again, thank you so much for your attention.
For any questions or issues you may have, please contact Avexia Diagnostics by email at info@avexiadiagnostics.com or by phone 5 days a week at extended hours, 8 a.m. to 8 p.m. Eastern Time at 888-852-2723. Thank you, Dr. Burnett, for this informative presentation. Thank you all again for joining us for this webinar event. Until next time, from everyone at Avexia Diagnostics and EPISeek, stay healthy, stay safe, and we wish you all the best on your pathway to wellness.